All right, I'm going to talk to you guys about the second assignment, the LeafPak simulation stream study. This is on the Google Classroom, so this link on your paper copy is in the classroom. If you just click on the stream study online, or here's a link for the LeafPak network simulator. So click on that, and it takes you to the following page. On your copy, you're going to select a study stream. Now there are 4 in total. That is a lot to do, especially when you start one. You'll see that it does take some time. So work with a partner, maybe do one and your partner can do the other. So you are going to choose between stream A, if you want stream B, stream C, or stream D. And you're going to share that workload with your partner. Noticing then if we were to do a bit of stream A together, it does a sketch of the stream for you, which is what your assignment asks for, is a sketch. If you look at your sketch, it doesn't look exactly like the picture because we only need certain aspects, which if we scroll down, it gives us a bit of a key. It says if there are any pools, it gives a swirl. If there's riffles, we'll notice a little more jaggedness in the water. And if there's runs, it shows it with the lines. So in my sketch, I have runs, I have riffles, and I have a pool. So in your copy, and you can even do it online here, I'm going to give each one of these for a click because it's got all 3. So in your paper copy, you're going to check mark pool, riffle, run. Now take a look at the stream bank. Does it have many trees, some trees, no trees, grass only, or pavement? Well, I would say we have many trees. So you're going to click off many trees on your paper copy. You're going to slide over to the second section. So notice on your paper, you can click over to 2. It gives you a new page and it talks about what's in the stream. Are there leaves, cobbles, woody debris, or plant roots? Well, there is a lot of plant roots and there's some cobbles. There's some rocks over here. The leaves that are in here, like if you look at the bottom, this is a LeafPak. This is what's going to be used to collect samples of what's in the water. So this is not part of just like the floating leaves. So I'm not going to click off leaves. For algae, is it light cover on rocks? Is there a thick cover on rocks? Or is there a thick cover on rocks, clumps floating on the surface? If you look at the rocks in the lake, there's only a little bit. So if you notice on my mouses, there's a little bit of green patch here, a little bit of green patch over here. So there's a light cover on the rocks. Is there fish, beavers, signs of trash, or drainage pipes? There are fish, there are beavers. So you're going to click off that on your paper copy. That part of the section for your habitat is done. What you're going to do is you're going to run a sample, but you have to stay consistent between all 4 habitats. So you get to choose on the amount of sunny days. If there are few, if there are many. So I'm going to put many, and I'm going to start, and I'm going to watch what happens. So if you look, notice how my fish population increased. And it stopped. 3 weeks went by. It brings up this LeafPak sorting tray. So what you're going to do, this just shows what was collected in that stream. You're going to click and you're just going to move the leaves out of the way so we can uncover. Oh, that leaf doesn't want to move. So we can uncover what was found in the stream. Okay, there's still a couple more leaves, but it's getting hard to move them over. Okay, good. So you're going to click over to the macroinvertebrates. You're going to use your dichotomous key that you used from the day before, and you're going to find what organisms you have. So for example, if you look at this big lobster looking thing, if you look on your dichotomous key, it's going to take you to a crayfish. So you're going to move this object into the crayfish zone. It gives you a total rating of 3 and it's somewhat sensitive. So if you go back to your paper copy here, you're going to click off on crayfish. So crayfish is present. Gives you a score of 3 for that organism. And it is somewhat sensitive. So we're just going to put S. Okay, let's go back. Let's see what else is in here. So we have a shell. We're going to look at your dichotomous key. I got to bring mine up here. Right, on my list, I'm going to look at the shell, and it looks between like a limpet or a freshwater mussel. So I'm going to go, and I have a section for clams and mussels, so I'm going to put it over there. And it is somewhat sensitive with a score of 3, so you're going to mark that on your sheet. Oh, that's supposed to be 3. Okay, somewhat sensitive. You are going to continue this until all these organisms are gone. The one I just picked up, it has 3 pairs of legs, so 6 in total with 3 little tails. So if I go to the dichotomous key, 3 pairs of legs, it has 3 tails, so I'm in this section. Kind of looks like a mayfly larva. So let's go, and it's not on this screen, so you might have to move over. Oh, here's mayfly. Let's see if it fits. Oh, it didn't. Or sorry, it didn't. So that's okay. Your screen will already tell you, nope, not it. Try again. So I'm going to go back and look at my dichotomous key and see if I can figure out if it's not the mayfly. Then maybe I'm getting the tail wrong. So just because of my dichotomous key and what this lab is using, this might be considered then a tail that's not obvious, whereas this object looks like it has the very distinct 3 tails. This one actually might be the mayfly. Oh, and it is. So you can put in your paper copy 12 and sensitive. That one that I picked up probably has a not obvious tail. If I look at the dichotomous key, there is a good chance that this is the the dragonfly larva. So I'll move it over. And it is. So I'm going to fill that in the sheet. You are going to continue to put these organisms into their section until all of them are gone. Once complete, you're going to get a score total, which the nice thing is about this lab is that it'll do it for you. So you can continue that until you get your final score, which will be done and calculated here. The last part, the tests. You're going to go to the chemistry section and you're going to run your tests. Oh, okay. For some reason, oh no, here we go. Okay, so I had to click over the first one, air temperature test. Step 1, read the thermometer. So I'm going to click on it, let it load, do its thing. It says look for the number that has the green background, which is 22. So you are going to put in the temperature of 22 degrees Celsius. You're going to complete that for the rest of the factors. So we're going to look at 2. You're going to do the same thing. 3, the pH test, collect a sample. Add the test tablet. And the match the color, which is just like we did in the lab on Tuesday. And it looks like that pH test, when I scan over, is a 7, which means that pH is excellent. So you're going to put 7, and that is excellent. Cool. Finish it up with nitrate, turbidity, and dissolved oxygen, and you have completed stream 1, or A. Then have your partner do another one like B, and then there's also C, and there is D. Once you're done all 4 of them, they'll all give you different ratings. You'll figure out what the stream health is and score it, whether it is good, poor, excellent. And then once you have all the answers from your partner and your work, you're going to answer the following questions. Is there a difference in water temperature between the stream? Why might streams C and D have lower dissolved oxygen concentration than streams A and B? And we talked about the importance of dissolved oxygen. On Tuesday, we kind of read the article about all the fish deaths in Alberta. All organisms that need that rely on respiration need oxygen. If the oxygen is not present in the water, things start to die. Number 3, speculate on what could be leading to the different pH readings in each stream. 4, what might be causing high nitrate readings in stream D and in C. And what are some reasons for the increasing turbidity in streams from A, B, C, and D? What factors might explain why there are fewer fish in C and none in D, and compare to the healthy populations in A and B? For question 7, now I look at this now, you guys can actually just skip that one. Don't do it. And which macroinvertebrates disappear first as the water quality declines? Number 9, without knowing the chemistry of a body of water, what would you conclude given the following macroinvertebrate community sample? So if we were just to go out to any kind of lake, pond, stream, wherever in Saskatchewan, take out a sample and seeing that we had maybe some riffle beetle larvae, some stoneflies, mayflies, fish flies, dragonflies, scuds, without actually looking at the other biotic or abiotic factors like air temperature, pH, nitrate, turbidity, dissolved oxygen, what could those organisms tell us about that stream health? And lastly, what would you conclude about a body of water with the following: scuds, leeches, midgeflies, blackflies, planaria, and aquatic worms? You don't have to calculate the PTI rating, but take a look on the dichotomous key and look to see where are they at in the high tolerance, low tolerance, somewhat sensitive tolerance to low oxygen levels. After that, you are done this assignment.